everybody, I'm Tall.
And I'm Small.
And today we're going to be doing Khan Academy's MCAT Prep Biomolecules, Kinetics, Enzymes. I had to mess it up eventually. Enzyme Kinetics questions. All right, let's jump right into the very first question. Given that delta G for the reaction SP with the double arrow is negative in the direction of S going to P, reaction equilibrium favors the formation of which substance?
So I'd argue that this is not even biochem.
Oh, really?
Yeah.
What would you say?
This is right back to Gen Chem and OChem and those kind of general principles.
Okay. So, the answer choices are the reaction does not proceed in either direction. P, being the product, the reaction proceeds equally in both directions, or S for the substrate. So, it's saying that the reaction, so delta G of the reaction is negative in the direction of S to P, which I remember delta G being negative is favorable. That's what you want. So the reaction equilibrium favors the formation of which substance? I would have to assume that it's P.
Yep.
Because if you have delta G going from S to P, then negative from S to P, then P has to be the more favorable product of that equilibrium. And sure enough, it is. Awesome. Next question. Okay, how would the addition of a catalyst to the reaction SP, once again with the double arrow, affect the difference between the free energies of S and P in their ground states? Delta G? I don't know why they wrote it like that. Okay, so if you add a catalyst to the reaction, and once again they're talking about the reaction where they're in equilibrium, how would that catalyst affect the difference between the free energies of S and P in their ground states? I'm tempted to say that it wouldn't change.
That's what I would say.
Okay.
I don't really know what they're asking here.
Yeah, but you would typically think, well, I mean, we've talked about it before, that a catalyst doesn't touch thermodynamics.
Yeah.
So I'm going to take a wild guess at it. Yeah, we
Well
got
done.
it. Very strong work. Okay. Next question. Oh boy, this is going to be a mouthful to read. I recommend all of you go on and just check out these questions for yourself because this is going to sound messy. For the enzyme catalyzed reaction E plus S, double arrow, ES, double arrow, E plus P, meaning an enzyme substrate come together and make a product. What equation defines the rate at which ES, the enzyme substrate complex, is formed? Is it K1 times concentration of the substrate, K1 times the total enzyme minus enzyme substrate complex times the concentration of the substrate? Is it K, the reverse rate constant, K minus 1, times the concentration of the substrate, or the reverse reaction rate constant, K minus 1, times the concentration of the substrate times the concentration of the enzyme substrate complex minus total enzyme concentration? Good Lord, that was hard to read. Okay, here. So what they're basically asking is, how do you define the rate at which ES is formed?
So
Go for it, because I have no idea.
yeah, so I don't remember probably more of this than I should. But what I do know is that, K1 would be in the forward direction, and K negative 1 would be going backwards. So I think I can take out the K negative ones.
Yeah, and isn't there something about when you're looking at equilibrium in this very classic way of describing the enzyme substrate complex? Isn't there something about that second set of double arrows that the enzyme and the product will not want to reform typically to form the enzyme substrate complex again?
I think that would probably change based on reaction.
Yeah. I mean, yeah, it would have to. I remember somewhere though them saying that.
So what they do to make math easier for us in biochem is that you can say K sub negative 2 is basically 0, and then you can go from that point. So is that what you're thinking?
Yeah, that's what I'm thinking.
Okay.
So in that case, let's really just try to look at these definitions. So they're so if we're assuming that the reverse is probably out of the question, then we're looking at either the rate constant times the substrate concentration, or the rate constant times total enzyme minus enzyme substrate concentration times substrate. So we want to know the rate at which ES is formed. I'm thinking that it's going to be the second one, the longer one, because that's saying that if you have total enzyme concentration, so the total amount of enzymes, you subtract the amount that are bound to substrate, that's going to give you the amount of free enzymes times the concentration of substrate that's there times the rate at which the reaction is occurring.
Yeah, and I think that sounds like it might be the right answer. I think I probably would have gone with K sub 1 times the concentration of S, because it looks like a rate constant that not a rate constant, but a rate equation that I've seen before. So I probably would have gotten that wrong.
Okay. Well, do we know if this is right? Oh,
Good
nice
job.
work. You got it. 7 more. Thanks, Sal. Awesome. Okay, graphs. Here we go. How can the Michaelis-Menten constant Km be derived from this Lineweaver-Burke plot? They give us a very standard Lineweaver-Burke plot, and they want us to derive Km. So I'm going to say real quick, because I know you know this stuff. Km is the substrate concentration at 1/2 of V max when you're looking at a Michaelis-Menten curve.
Yeah.
So to find 1/2 V max, they do show us that 1 over V max is the y-intercept for a Lineweaver-Burke. So I have to assume that it's going to have something to do with the y-intercept. Let's look at the answers. So the possible answers are -1 times the x-intercept, the slope times 1 over the y-intercept, 1 over the x-intercept, or the slope. So which one is Km? So based on what I said, the only one that includes the y-intercept in there, which they said that 1 over V max is the y-intercept, and we know that Km is the substrate concentration at 1/2 V max, then it would have to, well, just based on y-intercept, the only one that includes it is the slope times 1 over the y-intercept. Are you having any thoughts based on what you see?
I am not.
So if we had -1 times the x-intercept, do you remember what the x-intercept means in a Lineweaver-Burke?
I'm trying to think of which graph I am more familiar with, and this doesn't remind me of the one that I'm really comfortable with.
Typically when I see Lineweaver-Burke, it's always talking about competitive inhibition or something like that. So that I'm used to comparing different enzyme or different an inhibitor based on the normal enzyme function by looking at a Lineweaver-Burke.
Yeah.
But not really just looking at it to interpret what the Michaelis-Menten plot would look like. I would take this as a normal MCAT question, and I would say that because they're pointing out the 1 over V max is at the y-intercept, I have no idea really what the slope would be here. It's 1 over V naught, 1 over concentration S. I'm going to go with slope times 1 over the y-intercept because they talked about V max there.
So I know when I'm given, multiple lines, how to compare them and talk through what that means. But to be completely honest, yeah, I don't know how to calculate the Km from just one line.
I think the lesson to be learned here before we hit check is that everyone needs to really know those Lineweaver-Burke and Michaelis-Menten. You are almost guaranteed to have it on the MCAT. Let's hit check. Oh, and it was right. Okay, screw that, people. Just go with your gut. Okay, next question. Next question. Okay, question number 5. If the enzyme catalyzed reaction E plus S yields, or double arrow ES, double arrow EP, same as before, is proceeding at or near the Vmax of E, so when the enzyme is completely occupied with substrate, what can be deduced about the relative concentrations of S and ES? So the enzyme substrate complex and the total concentration of substrate.
So this is a concept question about enzyme kinetics, basically, that it is very basic, so hopefully I don't mess this up.
Okay.
But from what I remember about in biochem is that anytime I was approaching Vmax, I had almost infinity or just large abundance of substrate so that my enzyme was working at the fastest possible velocity that it could, just chopping up or doing the chemistry it was supposed to.
Yeah, and that reminds me of a Michaelis-Menten plot that, if substrate concentration was on the x-axis, as you keep going more and more on the x-axis, you see that it reaches that asymptote up at Vmax.
Mm-hmm.
So I would assume that substrate would be abundant.
Yeah.
We'd have tons of substrate.
So you can go ahead and take out, concentration of substrate is vanishingly low.
Which is also a very weird way of describing it.
Yeah.
Vanishingly. You got interesting adverbs, Con. Okay, so then what about the concentration of the enzyme substrate complex?
So at its highest point, to me, makes sense if the enzyme is working at the fastest velocity at which it can do the chemistry.
Oh, yeah, because all of the active sites are occupied by substrate. Therefore, it should be at its highest point. You should have the most possible at that point. And sure enough, we got it right. Awesome. All right. Okay, next question. Question number 6. What relative values of Km and Kcat would describe an enzyme with a high catalytic efficiency?
Oh, these are my favorite.
Okay, explain to me what Kcat is because I kind of forgot.
Okay, so Km, so I always relate that to my work in the lab, and it reminds me of that BLI, biolayer interferometry, dissociation constant. So
Okay.
It kind of gives us the number to where if I have a high affinity, I have a low Km, so I dissociate less frequently. And then Kcat is catalytic turnover rate for that enzyme and substrate pairing. And so
Catalytic turnover.
yeah, and so I think of that, as silly as this is, a cat turns over things on the table quite frequently. That is what they're known for. If there's a cup on the edge, it's going to crash and fall and break. So if you have a high Kcat, you're very efficient with that substrate.
And that means Kcat is literally saying, like, how quickly does a substrate go into an enzyme and then come out as a product?
Yeah,
Like, how quickly does that actually happen?
Yeah, the turnover rate.
Interesting.
That's like I can, if I'm a car cleaner, I can turn over so many cars in an hour.
Okay. So you would want a high Kcat, right?
Perfect. So go ahead and take out 2 of those answers.
Okay. And then I understand Km pretty well, and I would assume that you would want you would want this enzyme to be working really well, and if you had a high Kcat, then it would take a long time for you to reach the point where you're working at 1/2 your Vmax, right? Because it would always be churning out products. So I would assume you would want a high Km?
So a high dissociation constant. Yeah, that would make sense. I would be able to let go of my substrate more easily.
Sure, yeah.
So let's go with that and see.
Oh, I led us astray.
So low Km. So I was thinking, see, I should have stuck with it. So BLI, biolayer interferometry, allows us to check the binding kinetics of some probe with some protein on it and a ligand to that protein. And so you want a low dissociation constant. You want quick binding. And really slow off, so the 2 proteins have a high affinity for each other. So you want a really low dissociation constant. So a high catalytic efficiency for an enzyme if low Km means I have a high affinity for my substrate, that does make sense to me. And then a high Kcat, I would turn over each of those, those chemical reactions quickly.
Okay.
And get through more of them.
And sure enough, that was the correct answer, low Km and high Kcat.
So if it helps you guys remember that Km and affinity are inversely related.
Okay, so a low Km, if you think about that on a Michaelis-Menten chart, that means that it's a low, it means you reach Km faster with less substrate, basically, right?
You reach 1/2 Vmax?
Yes.
Yeah.
You reach 1/2 Vmax with less substrate, which is weird for me to think in my head, but I guess I can understand it.
So So the thing about these plots, is that I always have to include inhibitors when I think about them.
Mm-hmm.
And so I don't know, thinking about just asking about how one enzyme would deal with its one substrate doesn't make me think I need to go to the plot and think about it in that way.
Okay. All right. Okay, next question. Question number 6 or 7. No, it's 6. No, it's 7. For the enzyme catalyzed reaction E plus S double arrow ES double arrow EP, what is the value of Km if the concentration of substrate equals 25 and the initial reaction velocity is half of Vmax?
Oh, giving it right to us.
I was about to say, there's wait, hold on. So the initial reaction velocity is half of Vmax, and they give us the concentration of the substrate.
Yeah.
Isn't Km just gonna be that?
Mm-hmm.
25?
So what they want people to say is mess up Km, and so some people that don't have as strong of a biochemistry background or something just have memorized, and good on them for memorizing it with no context, but that
Yeah.
Km is 1/2 of Vmax. Well, that's not necessarily true. If we read the axis, or the axes, if that's the plural, that Km is the amount of substrate it takes to get to 1/2 of Vmax.
Right.
So yeah.
So the value, what is the value of Km if concentration of substrate is 25 and the initial reaction velocity is half of Vmax? Yeah, I can't even think of how to say it otherwise. We got it right, and they gave it right to us.
Con, you're taking it easy on us.
You really are, Sal. Okay, with respect to the binding of regulatory compounds, what properties define an enzyme as being allosteric? Is it irreversible covalent binding of regulatory compounds, irreversible non-covalent binding of regulatory compounds, reversible non-covalent binding of regulatory compounds, or reversible covalent binding of regulatory compounds? That was a mouthful. Okay, first off, it's reversible, right?
Yeah.
Yeah, it's got to be reversible. You can take away the irreversible ones. And then allosteric, so it's binding to some other site on the enzyme. I couldn't imagine it binding covalently. You're talking about a bunch of amino acids. I don't think they're going to want to bind covalently to anything. So I'm going to go and say it's non-covalent.
Yeah, so that is what I would go with. My favorite example of this is actually a system I work with in my research. And so it's a intracellular delivery system called TATCAM. And so we've introduced this viral-derived ability to bring it. So there's a nuclear localization sequence that really helps us. Basically, it's just a cell penetrating peptide that helps us deliver a specific cargo protein into the cell. But the cool thing about it is we've also covalently bound our cargo protein to something called a calmodulin binding site. And then our TAT, the actual delivery method, is covalently bound to something called calmodulin, which is this awesome protein that allosterically changes shape in the presence of calcium, which is much higher concentration outside of the cell. So our cargo protein and TAT bind really, really close. In the presence of calcium outside of the cell, it gets delivered, and then inside of the cell, where there's a relative lack of calcium, that non-covalent bonding dissociates, and through a nuclear localization sequence, our cargo protein can make it to the nucleus, and because it's a transcription factor, do what it's supposed to do.
And that's where you guys really utilize the non-covalent bonding aspect of that.
Yeah. So the people before only used covalent, and it was getting caught in an endosome, and anyway. The really cool thing is that it is non-covalent and it is totally reversible. So you were totally right in answering it like an MCAT question. Very well done.
Nice. Very cool. All right, next question. Question number 9. Assuming all other reaction conditions remain constant and that the reaction is allowed to proceed to equilibrium, how would the reaction S double arrow P, an equilibrium reaction, be affected by the addition of an enzyme catalyst? Okay, so we have a substrate and product in, or rather two sides of a reaction. In equilibrium, you throw in a catalyst, what's going to happen? Okay, here are the answer choices. Both increased reaction rate and increased formation of product, increased total product formed, neither increased reaction rate or increased formation of product, or increased reaction rate. Okay, so you throw in an enzyme, you're at equilibrium, what's going to happen? Do you have any thoughts right off the bat?
Yeah, that increased reaction rate is crazy.
Okay.
Yeah. So that's will let us eliminate 2 answer choices.
Increased total product formed. I mean, it's an equilibrium, so I don't think it would be forming product. Yeah.
So even if it did, even if we threw this catalyst in and the catalyst was super efficient in going from S to P, in my head, because we're at equilibrium, using Le Chatelier's, if I'm increasing the amount of product I'm making, I would immediately reverse it, go back to the substrate.
Ooh, actually, I'm going to zag on you here for a second.
Oh, no.
So because of that,
yeah,
the catalyst is causing the reaction to happen quicker. Even though it's at equilibrium, it's causing equilibrium to just happen quick.
We don't. So the phrasing is we reach equilibrium quicker, but we're not changing the rate at which the reaction is occurring.
With the catalyst?
Yeah.
So the answer would be either neither increased reaction rate or increased formation product or increased total product formed.
If you're wrong, I'm going to be highly embarrassed.
I'm going to click increased reaction rate because it sounds right to me.
If you are right, I will be highly... Okay.
All right.
I'm nervous now.
We're going to click this on 3.
Oh, no.
I have nothing to lose. 1, 2, 3. Yes! Okay. Sorry for your eardrums, everybody.
I quit science.
I will do my best to bring that down. However, let the world know that when you add an enzyme catalyst to an equilibrium reaction, you're going to get an increased reaction rate. Okay, so let's look at the hints that they give us. Catalysts function by lowering the activation energy of a reaction. Catalysts do not affect the position of equilibrium. Catalysts do not affect the direction of equilibrium. The equilibrium reaction would proceed at an increased rate with the addition of an enzyme catalyst. So you would have the same equilibrium. Nothing would be changed there, except that when you're in equilibrium in the first place, you're constantly going back and forth, like Le Chatelier's principle. That would just happen quicker. I'm assuming you don't like that answer.
I don't like how it's worded, but that's fine.
Okay, next question. Last question. Question number 10. Molecule X is an enzyme inhibitor that reversibly binds to an enzyme at a site that is distinct from its active site. Molecule X must not be what type of inhibitor? Uncompetitive, competitive, mixed, or non-competitive? So it says very clearly in many places that it's not binding to the active site. And of those choices, we know that competitive inhibitors are the ones that do bind to active sites.
Not necessarily.
For the purposes of the MCAT,
You're
competitive inhibitors
Okay.
do bind to the active site. Therefore, I'm going to go with competitive inhibitor. It feels like the only one that makes any sense based on what they give us.
Didn't you just say that competitive is the one that does bind at the active site?
Yes. So it's saying that molecule X does not. Therefore, what? What must molecule X not be?
And then non-competitive is also one that I'm assuming we're going to say that all competitive bind at the active site. So non-competitive could also, but not always. So that is not the one that's being ruled out here.
Correct.
Okay.
This is also a slightly good example of fine dining and breathing, people. And we got it right. Awesome.
So do we want to quickly go over the kinetics idea behind competitive, uncompetitive, and non-competitive? A good way to remember it?
I would like to, but I didn't get a confetti party from Sal.
Because you missed one.
Yeah.
Okay,
but
fault.
Yes, go over the kinetics.
Okay, so I have a wonderful story from my biochem professor, Dr. Glenn Meads from Kennesaw State University. And he came up with this, and it's a silly story, but you'll never forget it. And if you understand it, you never have to memorize any of the little relationships, because you will just get how things happen. And so there's a little dog, and he wants to go to prom. But when he shows up at prom, he didn't have a date, so he can't get in the door. So he just sits there and he watches how all the couples interact. And so, and then he just takes notes. And so the dog is like a scientist watching how enzyme binding happens. And so the first one, we're talking about competitive. So there's a boy and a girl, and they're dancing. They're a couple, but then there's this really weird guy who doesn't really get kind of how the social things happen at the high school dances. And his job the whole prom night is just to run and jump in between dates and grab onto the girl and hug the girl and not let go. And so he does that to all these different couples all night long, and that is competitive inhibition. And he's getting right in front of the girl and latching onto her active site. If it's inappropriate, you'll remember it.
Exactly.
And so non-competitive, right, is the inhibitor that wants to bind to the enzyme substrate complex together. And so there's this other really weird guy who, maybe just happens to be like the twin brother of the other weird guy. But this weird guy only runs up and hugs couples together. That's his thing. And
that's uncompetitive?
yeah, that's uncompetitive. So he hugs E and S together, the girl and the boy together. And because he does that, they seem like they like each other more than they do. And so there is an seemingly increased affinity, and therefore lower KM.
Oh my God, that is so good.
Right. And then going back to competitive, because I didn't mention this, which is the one that just wants to bind to the substrate, because it seems like the enzyme, the boy can never get to his girlfriend. There is a decreased lower affinity and therefore a higher apparent KM.
Whoa.
Yeah.
This is blowing my mind.
Yeah.
Okay, I need some clarification on what happened to this dog.
Yeah, and so the dog is just taking really good notes this whole time, and he's figuring out how the interaction happened between the girl and the boy, and then the two weird brothers.
Okay.
And those weird brothers have a third brother, and he kind of likes to do what the first and second brother do. So he will hug either one of the couples, and so he just goes around making chaos for everyone. And that is the one that we don't really get questions on the MCAT, and that one is called non-competitive.
Hence the absolute drop off of the explanation of inhibitors at that point.
Yeah.
But that was actually phenomenal. Literally, that was so well explained. My only question is, why was a dog taking notes at a prom?
I don't know. My biggest question with that is actually that Dr. Meads is a cat person.
I know, right?
Yeah. So I don't know why it was a cat. Wasn't a cat.
Why wasn't it just a person?
Yeah.
We had to anthropomorphize a dog rather than just say, oh, it was just some other dude who's there.
I don't know. He's an MD PhD. He can do what he wants.
That's the rules, people.
Yep.